All right. I think we are. Yeah, we're recording. Okay, guys. Now we're on the dial. All right. Yeah, I agree. I think it is a good study guide too. I mean, that's yeah. So here's what's going to happen. After this thing uploads on my computer, I'm going to throw it up on the YouTube site. I think you guys at this point are able to find me on there. I know people are asking for my channel name. I don't know. It's Drew Mattmiller, I guess. I just type in Bio 137 Mattmiller, all my crap comes up. So that's how you find this, and it'll be out fairly soon, I hope. Okay, so let's go back to let me share my screen again. All right, so now we were talking about the simple squamous epithelium. Excuse me. We have to have a a one layered cell line here because we are diffusing gas. Gas cannot travel through a bunch of stratified, excuse me, layers. In the lung, if you have alveoli, it's had to be very quick, and we have to have very thin a thin barrier so we can exchange carbon dioxide and oxygen. Because if you think about it, this cell layer has capillaries in it, and inside the capillaries are the circulation. And this is where red blood cells will come in and drop off their oxygen and pick up their carbon dioxide to be exhaled out. So it's kind of cool. That's the reason why it's simple and not stratified. All right, so now we have an example of a simple cuboidal epithelium. All right, so when you think of cube, you think of kind of like a chalk of ice, right? Maybe like a piece of a square cell, like checkout, kind of like these look like more square-like. Now the little purple dots in there, those are going to be the nuclei, the nucleus. The nuclei is just a plural term for nucleus, right? All right, so usually around, let's just say yeah, I think we covered all that. Okay, so this is an example of the kidney tubule cells. So this right here, I think some of y'all even had the kidney tubule cells. Like this is what a nephron looks like inside of a nephron. This right here, this is called your lumen, which is pretty much the whole inside where we would have urine or blood flow coming. More likely it's going to be urine because it's a kidney tubule cell. So this is your kidney. This is the whole kidney tubule cell. Tubule. It's not a cell. I'm sorry. It's a structure. And this is where you would have. This is how you filter your urine. This is where you would have your urine or waste product in here. It will be flowing through. And then the function obviously is for absorption and secretion. So as urine or metabolic waste runs through these tubules, these cells, these simple cuboidal cells, will absorb and secrete different compounds depending on whether we need it or not. This would be bicarb for pH purposes, sodium if we're low on sodium, if we have too much potassium. We'll say we ate a bunch of bananas which are high in potassium. Your body does not want a bunch of potassium because it will stop your heart. So what happens is that we may excrete a lot of the potassium into these renal tubule cells for nutrition, otherwise known as urination. It leaves the body. So features to know basically is just the nuclei here, you know, and it's simple cuboidal. Check out these look like little ice cubes with dots in them. Maybe that'll help you remember them. Okay, and now simple cuboidal. I think the simple part, I'm going to draw your attention over here to single layer of cube-like cells. I think single layer, I think is the key term there. All right, next one, simple columnar epithelium. Now, simple, so I think we're going to say single layer, right? That's what we started off with, column-like cells, usually in a fairly in-tune layer, I guess is what I'm trying to say. These are going to be likely in your GI tract. Okay, as we can see here, the jejunum, which is part of the small intestine. So these are also part for absorption and secretion. And they're going to have features that we have pointed out here. Number one, that is a goblet cell, okay? Goblet cells are specialized cells that are involved in secretion of, I want to say, types of proteins. It may be pepsin or it may be an acid, hydrochloric acid. I'll have to Google what it may be. The goblet cells, I think, are part of what secretes your stomach acid. Something involved there. You don't have to know it, but you do have to recognize what goblet cell does. And then, of course, we have these oval-like shapes here inside the cells. Those are going to be, that's going to be your nucleus. Every cell is going to have a nucleus. And then, if I don't know if I can zoom in here and it helps, but if you see this layer right here, this is your microvilli. All right. So the interesting thing about microvilli, another goblet cell here, by the way. All right, goblet cell. This is kind of like the lumen here. So it's going to secrete into this cavity. This is where nutrients come in and everything, and it'll absorb in. Okay, so microvilli are what's really a structure. It's not. It's made up of a bunch of different cilia type structures. They're kind of like waves, like hairs that stick up, and there's a bunch of them, and they move together in succinctum, and that helps propel nutrients towards the cell layer, the cell surface. And when nutrients get towards the cell surface, it'll either grab them and absorb them in, or they'll pass them along. Microvilli are usually part of your GI tract. Cilia, I use the word cilia because it's hard to explain. Cilia is the same thing, but that's found in more of the epithelium of the esophagus, or the trachea. It's the trachea. And possibly the esophagus too, but I know definitely the trachea, where it'll help you filter the air that is coming into your lungs. This helps kind of when you cough up phlegm, I guess is the proper term. That is the microvilli kind of absorbing foreign bodies and propelling them out. They move in one direction, which is from bottom to top, which is supposed to bring it up towards your mouth, and you can either spit it out or you get rid of it another way. And that's just foreign bodies. Now, the reason why, the interesting thing is why a lot of smokers have bronchitis is because when people smoke, the smoke will hinder the cilia in the trachea and the esophagus. And what happens is you get a lot of bacteria and foreign bodies that make it their way into the lungs because of a lack of movement of the cilia. And that's how bronchitis, a lot of people who smoke are more prone to bronchitis and other pulmonary issues, is because the cilia are not able to work as actively as they normally would with somebody, a non-smoker. So it's kind of interesting. Back on the microvilli, it's the same idea here. They all work together to try to bring electrolytes or nutrients or sugars, anything, proteins, anything that the body may need, so it can come in contact on the cell surface and be absorbed into the GI tract. All right, so 1, 2, 3, simple columnar epithelium. Make sure we understand where we can identify microvilli, the goblet cell, and of course the nuclei. There's going to be questions on the nuclei, show nuclei or nucleus. Same thing. All right, so pseudo-stratified columnar. Now, this is what I think I just talked about. This is, this is out of a trach. This is the trachea. So number one here, there's your cilia, right? A little bit different, more hair, more longer than the microvilli. But you can kind of understand the wave feature that I'm thinking about. Think about it as like seaweed underwater, or, like when you see these pictures underwater of like the Great Barrier Reef, or you have these seaweed structures, and they just kind of float together with the current of the water. That's what I'm trying to explain to you guys, what cilia and microvilli do. And when you put all these different structures together, they can have quite an effect. All right, so let's see, pseudo-stratified columnar. Is there anything interesting here? I think it's going to be the cilia part, secretion and movement of mucus. All right, that's interesting thing with pseudo stratified columnar. You're going to find it in the trachea. And do we have goblet cells here? We do. These are harder to find. These are the still, you can see these little structures in here. Goblet cell, goblet cell, goblet cell, goblet cell, goblet cell. The circles are the nuclei. And you can kind of see where the cilia have branched out from the cell and are into the lumen. Okay. Let's go down to stratified squamous epithelium. And unfortunately, I don't think I can see my chat through here. So if you guys have chatted something, you might want to speak up because I cannot see the chat from here since I'm sharing my screen. All right, so stratified squamous epithelium. It's all about epithelium. All right, so I cannot think of anything that is interesting here to talk about other than it is the same thing we've been kind of covering. We have the nuclei here in the dark purple. Stratified, let's talk about that. Stratified is the key word here. So you see different layers of this, right? Lots of different layers. This would be something like out of like, here it's going to be the lining of the mouth and esophagus, okay? The skin, I believe, is stratified also. So let's see, many layers of cells. Cells towards the surface are more flattened and become more cuboidal towards the bottom and darker. So you can kind of see that. We have a flatness over here where these cells have been smushed down. See the little purple lines are the smushed out nuclei. And then as we go into the deeper layers, you can see how they are more rectangular, more columnar. Okay, not exactly like a flat layer either. They're more ebb and flow layer, perhaps. All right, and this is more for protection, abrasion of infection. This is like the quintessential epithelium purpose right there. All right, nothing else to see here on stratified squamous. All right, now I think we're done. Well, we're still doing epithelium. Okay, one more, sorry. Now, transitional epithelium. This is something that pops out in your urine sometimes. Sometimes if you were to submit a urine and you may have a UTI infection, if I see a bunch of transitional epitheliums, this tells me that you have an upper UTI infection or UT infection, urinary tract infection, UTI. And these are kind of interesting. That's probably about the only clinical indication that these have, because when you are under a bacterial infection, these will slough off. These will kind of fall into the urine, and then when you go to the ER because you need some antibiotics, I will look at these. I will look at urine underneath the microscope, and you could see a lot of these guys floating around, and that just tells me that you probably have a urinary tract infection that is, I guess at this point it's turned into cystitis. Cystitis is an infection or inflammation of the bladder, and this is kind of where these guys hang out. So, like I said, ureter or urinary bladder location. They do have somewhat of an irregular shape. I'm not sure what they quite look like. They're kind of more like a teardrop shape, almost more like a teardrop. Larger at the top, and then kind of it will kind of form down into a tail. And features to know, well, it's just the nuclei right there. And probably the purpose, right, and where they hang out is that if they hang out in the bladder and the ureter. Like, let's say if you have a stone stuck in your ureter, if you're able to produce any urine specimen, chances are it's going to be bloodied. And a lot of these transitional epithelial cells may present themselves in the urine. Okay. Well, and then we have this disclaimer. You will not need to know anything about stratified cuboidal or stratified columnar epithelium in this lab. All right, so now if we were in lab, this is where we would pull out some, and we kind of did this last week. I kind of wanted to have you all pull out a tissue and try to get it focused, and I think we did that. I came around and made sure everybody was okay, but if we were in lab tonight, this is kind of what we would do, and we can do this next week too. We could bring out the microscopes and do this next week, and or the week after that, or we could do this before the lab exam. The lab exam is going to be online, and you know, it'll be pictures from these different tissues. Okay, let me, I'm gonna stop this real quick. I'm gonna come back to the, okay, so we're good on the chat. All right, let me scroll down from 3.5 to 3.6. This basically tells you to label the different features of each cell. And we went over that. Okay. Now, let's go back on sharing the screen. All right, connective tissue. All right. So this is connective tissue basically is part of the extracellular matrix. And it's everywhere. It's like a webbing. Think of it as like a webbing, and it attaches everything to the muscle, to the bone. It keeps everything from jiggling around too much. You know, if we were to be running or doing some kind of exercise, connective tissue kind of holds things in place, and it's important, and they're everywhere. And we're going to kind of show a couple examples of what connective tissue looks like. All right, so visible cells and connective include fibroblasts, lymphoblasts, and adipocytes. Lymphoblasts, I'll be very interested to see what lymphoblasts looks like, because I think what we mean by that is lymphoblasts are probably going to be in the bone marrow. They're not going to be out in the connective tissue region. Okay, let's scroll down to areolar connective tissue. Let's start here. All right. So this looks like some like Jackson Pollock painting, you know, where somebody's just throwing a bunch of stuff on a piece of paper. Now, these are elastic fibers, and there's probably no way you're going to be able to identify a fibroblast. Okay. The reason why it's a fibroblast is because it has the dark nuclei. You see the darkness there? That's going to be your nucleus. This right here is a fibroblast. That's a nucleus. That's your nucleus. That's your cell. So there's your fibroblast. Now, what I meant to say is you're not going to be able to discern the difference between a collagen fiber and elastic fiber. So when you see like this would be either a collagen or elastic fiber. Up here is another example of a fibroblast. Okay, fibroblast and then the cell around it. It's really hard to see where the cytoplasm ends and where you know, the extracellular fluid begins. So hard to really point out what's what there. So this is going to be found areolar connective tissue, in my opinion, I think is what really thinks is going to be the lung tissue. But I think here they're underneath. We have most organs underneath the epithelial. Okay, so I think it will be a little bit everywhere. Okay, so and the identification here. Think of this term as like a crisscross. It keeps everything in line. So highlight the crisscross and kind of keeping everything together. All right, next one, reticular connective tissue. All right, these are dark stained, and I'm not sure why they stain so darkly. It may be the type of protein that makes up reticular. These are going to be a type of tissue that is found more internally. Okay, and this is going to be these are going to be like your, what's it say? We have the spleen and bone marrow and lymph nodes, and this is. Okay, what it's saying, lymphoblasts and there's no other brownish colored characteristics of this tissue. Okay, that didn't really help answer our question. So I guess okay, so we are in the bone marrow. That's why we have lymphoblasts. That makes sense. So there's not much to add here that I can think of with reticular formations. This, I think, was what would happen if you would have like a lymphoma. If you have a lymph node that is completely blown up, the reticular connective tissue, I think, would play a role in that. You probably want to clear that out because that's not where the pathology is going to be. The pathology is more likely going to be the non-connective tissue, unless it's like a sarcoma. A sarcoma deals with connective tissue. But in this case, I think the thing to point out here is that you're going to have these reticular fibers running across. See, like you can barely see it. Like that is reticular fiber, and all these little dark dots in here are going to be the lymphoblasts. So obviously we're inside the bone marrow where lymphoblasts will eventually mature to become lymphocytes, and they'll go out and fight COVID-19 or other viral infections, carry out other jobs. Like here's a reticular fiber. I cannot think of any other ones that I see. Okay. So dark stain, chances are it's going to be reticular connective tissue. All right, adipose. Adipose is is the proper term for fat cells. And as you can see, they're pretty much a cell membrane packed in with different fat compounds, okay, lipids, all kinds of different fats. All right, so they're generally large, the entire volume, which is made up the fat vacuole. That's what I was looking for. And the thing is, it's really hard to see the nuclei. Okay, I would think that right there is a nuclei. The arrow here is a nuclei, but the take home here is basically when you just see like an empty cell with the cell membrane and you're looking at fat. This is going to be for energy storage. This is going to be for muscle energy that you need, or if you undergo what is it, a fasting situation, like this is where your energy source comes from. Depending on whether you're male or female, will show where a lot of these are deposited. Fat cells are deposited. Females and males deposit fat at different areas in the body, and this is kind of where you can where you can see what it looks like underneath the microscope. To the naked eye, it looks like just a bunch of yellowish mush. We've probably all seen that, where fat is deposited in and around different areas of the body that you have to pick through. Okay, next is dense regular connective tissue. Well, I think this is going to be dense regular connective tissue is going to be your tendons and ligaments, okay? And these dark stained purple areas around here, this is going to be your fibroblasts. Fibroblasts are the ones that pump out collagen and other connective fibers to produce these, okay? Like if you have a tear, you tear your ACL, they're going to obviously do a surgical connection, but what really heals the ligament or the tendon is going to be the fibroblasts, right? If you have, like, let's say, the interesting thing with fibroblasts is like any kind of wound healing, they all come back as fibroblasts. For example, the big thing about this right now is when you have heart failure. Not only heart failure, let's say if you have like a heart attack, right? So if you have a heart attack, your cardiac cells, chances are they did not receive enough oxygen or nutrients from a blocked artery, so therefore they die. And when they die, the cells that replace them in the heart, it's not new cardiac cells. Some are, but chances are they're not. But what it really is, it's fibroblasts. And so all of a sudden, remember when we were talking about this heart beating with the cardiomyocytes? When cardiomyocytes die, cardiac cells die, they get replaced by fibroblasts. And fibroblasts don't beat, okay? They're just there. They're just there to produce a bunch of collagen and plug up whatever wound was there. So all of a sudden, you're losing this synctum, this ability to contract and push blood out of the heart, and therefore your heart becomes slightly weaker after every insult to the heart, whether it would be a cardiac myocardial infarction or some degree of heart failure, or you just have a chronic blockage. So fibroblasts come from everywhere. They're really just there to plug up the wound but they really don't add anything important. Same with kidney cells and lung. When you have more of what we call a fibrotic organ, it really takes away from their function. So right here is the dense regular connective tissue. And these are going to be involved in ligaments and tendons. So it's kind of have this wavy-like feature. That is something I want to point out here. and they're densely packed. They're supposed to be parallel, but like I said, this is more like a ebb and flow deal. Not as much as the pseudo-stratified, but definitely you can see some indications of a wave-like structure there. Okay, now we have dense irregular. This was regular. This is irregular. Okay, so this is, I'm trying to think, I have to cheat here. This is, okay, this is the dermis of the skin. Now, I don't know what else to add other than it's just, it is not very uniform. That is more like it just speaks to irregularness, right? There's nothing to add. It is going to be seen in the skin, and this is going to help with impact or stretching, and it kind of has this overall blotchy appearance, I guess, is how you identify this. I have no interesting takes from dense irregular connective tissue. If you were to recognize it, then you know that this is dense irregular. You'll never see anything else like it. I guess it's left me speechless. Okay. Elastic connective tissue. Well, let's think about this. It's just what it sounds. It can stretch. This, I don't think stretches as much. This can, because it's more organized. All right. Elastic connective tissue. When I think of elastic connective tissue, I think of arteries and arterioles. And I think, okay, this location is the aorta. So now this is going to help. This helps to keep your blood pressure low. All right. When we have high blood pressure due to old age, that's what happens when we have arterial sclerosis, which is mean the hardening of the elastic connective tissue. When we are young, this type of tissue is what we call, it can compensate. And what I mean by that is that when the heart pumps blood, it will expand to accommodate that massive influx of blood, and then it'll kind of pull back, and that helps push the blood out into the body, the systemic circulation. So all of a sudden, the left ventricle will contract, which the left ventricle goes to the whole body, and then after the blood leaves the heart, the first structure it goes to is the aorta, which is kind of like this big hook to the top of your heart, and it hooks down. And we have all this blood kind of going to the aorta, and how the aorta will compensate this is that it'll expand a little bit, and that kind of helps keep the pressure low. We don't want something that's real stiff, because that will increase the blood pressure, and that's not good, especially for our kidneys. And what happens is that it'll compensate, get larger, and then it'll kind of recoil back to normal, and that helps push the blood out into the systemic body. And the interesting thing about, and what happens is when we get older is kind of when we have, I believe it, atherosclerosis is the plaques that are deposited. Arterial sclerosis is the sclerosis or the hardening of the elastic connective tissue. So there is no more compensation when it comes back and forth. It's just stiff. And when you have stiff when you have a stiff aorta, that will cause your blood pressure to increase, mostly seen in the older age groups, just because the fibroblasts are not making nearly as much elastic connective tissue, and things die off, and it increases our blood pressure, which can cause, if we're not careful, it'll smoke our kidneys if we can't take care of it and get under control. All right, so I'm not sure if there's any nucleoli around here. It looks like we have collagen and elastic fibers right here that are darkly stained. I don't think anything else. There's no nuclei there. So we've got taking a good look at elastic connective tissue. We kind of understand what it does. Hopefully that'll help you remember it come test time. All right, so now we're scrolling through some things that we would do if we were in class. Same thing we kind of did last week. Be able to label the fibroblast nuclei, collagen, elastic fibers. We've pointed those out. These are all the same. Label the adipocyte nuclei if you can find one. that'll help you study when you find, the good way to do this, like I said, is do a Google image search and then see if you can pick out these features on the image itself. That's kind of how you learn. The goblet cell is the big one I've seen in the past, is trying to identify the goblet cell. Okay, now look, we even got a table for you. So this will help you fill out the table, and this is kind of like your flashcard, right? Once you have the pictures, you can go through here and label the flashcard, or label it if you do flashcards. That's, some people are flashcards, some people aren't. I was never a flashcard person. I know a lot of people are, and this class is set up for a lot of memorization, just like any other introductory class. But you don't want to memorize it too much, because you have to understand why we're learning it, like the function of it. That's what I'm trying to bring in, like some real world examples, kind of help why we're learning this this type of information. Okay, so we have the same thing here with connective tissue, and I think that's it. Okay, I'm going to stop this and go back to the chat. So what do we think about all that? That's a lot of material. Tissues is kind of up there, probably the hardest lab, if not the second hardest lab. Are we okay? Do we have any questions? I guess did everybody just leave? Okay. Yeah, Lindsay, the test is, I think I'm going to do an online test. I mean, I know we're in person, but I can't, everybody's going to show up. We're only supposed to have an X number of people in there at a time, and I don't think I can accommodate everybody with the test. Now, so I'm probably going to proctor it. I'm going to probably use the Proctio system. Now, if you don't want to do the Proctio system and you want to come in person, then you bring your computer, bring your charger, and I'll be your proctor. We'll probably do something like that. I think I got to look at the syllabus. I'm sure it's coming up, and we'll do it. We'll do it. And I need to send out the email how you can load up the Proctio on your computer for some of you all who are not familiar with it. But yeah, I think I'm going to do it online. I'm going to have an option, basically. If you want to do it in person, you can. If you want to do it online, that's fine. Okay, so are we okay? No, nothing else? I'm going to stop this recording.